Right, kia ora koutou katoa, ko Mick Toku ingoa, nā mai haere mai. Welcome along to our Friday afternoon Genomics Aotearoa seminar series. I'm Mick Black. I am one of the co-directors of Genomics Aotearoa. I'm going to host today's seminar. So hopefully, whatever part of the country you are zooming in from, you are not faring too badly from the weather. You may note that behind us, Dunedin is actually turning on not a bad day, which is rather unusual. We probably only get a good day when everyone else is having a bad one. Um, and if you're a latecomer, you also missed Gemma and I trying to wave to each other between our buildings, which was one of the highlights of my day so far, actually. So it is my great pleasure today to introduce our seminar speaker, Professor Jemma Geoghegan from the Department of Microbiology and Immunology, just behind me, behind my screened off window there, at the University of Otago, where she is the Webster Family Chair in Viral Pathogenesis. Many of you will know Gemma already. She's a Rutherford Discovery Fellow. She holds a joint appointment with the ESR. She played an integral role in helping with New Zealand's response to the COVID-19 pandemic. And during that time, she was an amazing scientist, and she was also an amazing science communicator. So I still fondly remember those few TV appearances back then, and all the amazing work that you were doing with the ESR team. I've actually known Gemma for quite a while, perhaps longer than Gemma even realizes. I think my first memory of Gemma was her giving an amazing talk at a Genetics Otago meeting somewhere down the road by the gardens. I think you won a prize for the best student talk that day when you were still Hamish Spencer's PhD student in zoology. So since then, Gemma has had a pretty amazing career, and I am really looking forward to hearing her talk about it today. So Gemma, thank you very much for agreeing to talk with us and share your research today. And I will hand over to you and disappear.
That was great. And yes, I do remember that winning that prize, which was very surprising because I was trying to speak about something that I didn't really understand. But so thanks for the introduction, and kia ora koutou. Thanks for coming today on a Friday afternoon, and I'm hoping you're all dry and safe. Today I'm going to talk about genomics-informed diagnostics for infectious disease, and I'll probably only mention COVID probably once. So and I'll try not to talk too much about that. So let's just start this. Okay, so in 2020, when the rest of the world was kind of trying to grapple with a new pandemic, a novel lineage of avian influenza virus also emerged about the same time. And this lineage of H5N1 subtype had adapted. And it adapted to both expand its host range and therefore geographical range. And it's now spread to all continents besides Oceania. And it's been killing millions of birds worldwide, with multiple spillovers to a broad range of mammalian species as well. But most concerningly, I guess, for human health and public health, this highly pathogenic flu has acquired the ability to transmit between mammals for the first time. It's spreading between dairy cattle with an extremely high attack rate, and is now spread across farms right across the US. Spread, all 50 states have reported cases on farms now. And so more recently, we know that this virus has spilled into pigs. However, underreporting, especially in recent months, in this US administration, has meant that we don't really know the true extent of this virus in pigs. And there's been many spillovers to humans as well, largely limited to those humans that have been working with infected animals. But with bird flu, at least, which seems really, really scary, we know the virus, and we even have really effective vaccines for it. So what happens when people start getting really sick and we don't know the cause? Well, this, of course, happened in late 2019 when clusters of cases of this undiagnosed respiratory disease were admitted to Wuhan Central Hospital. And fortunately for the rest of the world, Chinese scientists were able to use genomics, or metagenomics in this case, to rapidly find an answer. And back then, when Twitter was still cool, this meant that this full virus genome, within days of scientists finding this, was publicly shared. This genome enabled PCR tests to be designed and vaccines to be developed within weeks of this being shared. So, and this is kind of the situation where a lot of countries are going, where genomics-informed diagnostics is embedded in public health. However, in New Zealand, we're quite far away from this happening. It is a work in progress, and we have various projects kind of trying to implement this and in a research setting. So perhaps ask me in about a year if this is how this is going. However, in the meantime, though, we have animal disease, and there's a lot of it. So while we anticipate the arrival of H5N1, which will likely wipe out a lot of things anyway, only half kidding, we can help by using genomics to better understand infectious diseases in both wildlife and domestic animals as well. So in recent years, we formed really excellent collaborations, particularly with the Department of Conservation, MPI, disease incursion investigators, and other scientists, particularly those sort of at the forefront in industry, for example, in the Cawthron Institute. And we've been working with, especially on taonga species that are grappling with many infectious yet seemingly cryptic diseases to try and understand the causes of them. And what genomics helpfully gives us is a list of suspects. And in most cases, that's great. That's what we want, is trying to understand the cause. But a lot of the time, this list can provide a lot more questions than answers. And hopefully that's kind of the vibe that you'll get today from the seminar. So let's start with one disease that affects perhaps our most iconic species, the brown kiwi or Rowi. These Rowi suffer from a dermatitis disease, which can be really, really severe, affecting their skin. And it's been known to cause these outbreaks since about 2013, but no known exact cause has been found yet. So we worked with the Department of Conservation and a few vets at Massey University to try and understand what might be causing this disease. So we used a genomics approach or a metagenomics approach to try and understand this, which means bulk RNA sequencing, short read shotgun sequencing. And so just a snapshot of what viral species is in these samples, comparing across the disease range, as well as a few healthy controls as well. We found 3 different virus species. Interestingly, you see this pattern where this viral species in the Flaviviridae is really highly abundant, but really only limited to these mild cases, quite surprisingly. Then there's a few other species in the coronaviruses and picornaviruses as well. So looking at the evolutionary history of these viruses to see what kind of species they are, to see whether or not, they're important for avian hosts or related to other viruses that cause avian diseases. They are all related to other viruses that cause infection in avian hosts, but not necessarily cause disease. However, this one that we're particularly interested in, this one in the Flaviviridae. So flaviviruses involve things like dengue virus and yellow fever and things like that. You've probably heard of West Nile. This virus, let's just zoom in on that part there, is a sister taxa to a virus that's called bald eagle hepacivirus. So this affects bald eagles, yet another very iconic species in the US. And this causes really severe neurological disease in bald eagles. When I say it's sister species, that's the closest known relative, but at an amino acid level across the whole genome, it's only about 50% similar. So it's still really, really, really divergent. It's just that we don't know anything in between them. So this is a virus that's present at really high abundance only in mild cases, which is quite intriguing. And if we were to assume that this might be the cause, then perhaps more severe cases have rid themselves of the virus and have a secondary infection. So we didn't want to limit it to just viruses. We wanted to look at the whole sort of infectome, if you like, so all the bacteria, fungi, archaea, parasites, and we kind of look at this pattern among these different sort of family levels of these different organisms. we find really significant clustering of severe cases in particular. And when we look at the abundance of everything that's in here, apart from viruses, we see this really, really highly abundant nematode called Eucoleus in only severe cases, including in the skin lesion biopsies as well. So when we look at this species, we were able to recover quite a few genes and look at the evolutionary history to see this looks like potentially new species of nematode in these kiwi. And it's really, really highly abundant in these severe cases. And so, we kind of might hypothesize that potentially the Hep C virus is important for causing initial infection, and then sick kiwis get infected with a secondary pathogen, allowing the nematode to cause really severe disease. And this is all very arm wavy, and this is what genomics has told us. So there's actually a lot more questions than answers, but at least we've kind of got a list of suspects that we might need to look into further. And so there's lots of challenges with diagnostics in wildlife disease using this approach, using a genomics approach. Basically, we have none of these things, which is really important. For example, in the case of kiwi, those health checks are done annually. So we don't understand the epidemiology of the disease at all. And they're only caught when they're checked for health checks. So it could be at a severe stage of disease and we don't understand that progression. We don't really understand in a sample that's giving us a whole kind of infectome or microbial community what is causing a primary infection versus a secondary. We cannot do challenge studies in endangered wildlife, especially in kiwi. I don't think that would go down very well. There are no cell lines or model organisms to try and do in vitro studies that would, the closest model organism for birds is chickens, and New Zealand birds are quite far away from that. Most things don't grow in chicken cell lines. We also don't know much about the host, in comparison to human health, we have lots of model organisms and we know lots about the human genome. But for most of the time, there's no host genomes, or we don't know much about the host genetics. So we don't understand the combination between what the host genetics is contributing to the risk of disease. And it's when we're understanding a microbial community, most of it is probably not contributing disease. So understanding the normal microflora versus what is pathogenic and what makes potentially a normal microorganism become pathogenic is something that we have no idea about in wildlife. So all of these things are really, really, really challenging. And another kind of example that fits really into this can of worms is a respiratory disease in yellow-eyed penguins. So this is really live. So there's these the yellow-eyed penguins, or hoiho, are really endangered. Apparently the rarest penguin in the world, and they exist mainly around Otago. They can exist up to Banks Peninsula and down to Aotearoa on the mainland, and that encompasses the northern population, and the southern population exists on the subantarctic islands, mainly Enderby Island and the Auckland Islands and Campbell Island. So the northern population in about 2019, they first started getting a really horrible respiratory disease, which affects really young chicks a couple of days after they hatch. And it has a 100% mortality rate, and about a quarter of the chicks get this disease every year since for the last 5 years. So it basically is a hemorrhagic disease. It fills their lungs with blood, and they die quite instantly. And so histopath kind of suggests a potential viral origin, but it was really important to try and understand what was causing this disease very quickly. And the only potential pathogenic thing that we found through using metagenomics was a virus that we have termed yellow-eyed penguin gyrovirus. This is in a clade that, or in the same genus as other gyroviruses that can cause disease in birds. There's also gyroviruses that are in completely healthy birds as well. The only gyrovirus that we really know anything about is chicken anemia virus because the poultry industry vaccinate it, and it sort of behaves in a really similar way to this yellow-eyed penguin gyrovirus. So, and having looked at, the whole sort of infectome, all of the microbial community, we're really quite confident that this virus is probably the likely cause of this disease. However, so like I said, this is the sort of distribution of Hoiho, but the disease is only in the northern population. But having now sampled Hoiho from the entire host range, we know that the virus is everywhere. Completely healthy Hoiho in both the subantarctic and the northern population can test positive for the virus and have an asymptomatic infection. However, the disease is only in the northern population. So at first we thought potentially there is a lineage of this virus that is causing disease. And we have now gone back through about 20 years of postmortems, mainly that have been done at Massey University, to test old samples for the presence of this virus. And we know this virus has been positive, has been around in yellow-eyed penguins for as long as we've been collecting samples, which is about 20 years. So we've just started this, but we've sequenced lots of gyrovirus genomes. This is a virus genomes across those 20 years, and we're currently undertaking a lot of analysis or time-scaled phylogenetic analysis to try and understand these dynamics. But just really preliminary tree here, which shows sort of superimposes disease and non-diseased. There's lots unknown as well, especially in the subantarctics. But you can see there's no obvious clustering. So, and I mean, we're going to look into this further and subtle amino acid changes to see whether or not there are lineages that potentially result in severe disease. But we don't think that it's necessarily the virus that actually, is actually the thing that's actually causing, is a decider whether or not it's pathogenic or not. Although we think that the virus is causing disease. So this has opened up a huge can of worms, and thanks to Genomics Aotearoa for funding another side project where we decided to sequence the host genomes. So we managed to sample about 250 Hoiho, so about half from the southern population and half from the northern population, both with disease and non-diseased and positive for the virus and negative for the virus in both populations. And so we're trying to understand, basically it's a penguin GWAS, trying to understand genomics, if there's a genetic risk for disease in the northern population. And we're still working on this, and this is a collaboration with us and Joseph Gulen, and we're looking at all of these genomes and starting to understand that actually this northern and southern population, although they've been known to be separate populations, we didn't understand the true extent that they were separated until now. And in a two-dimensional plot, this is what it looks like. This is what the sort of SNP analysis looks like, where we find really distinct clusters of this populations. And really surprisingly, and I think it surprised everyone, is that in the southern population is split by Campbell Island and Enderby Island. And we find that there is literally no migration between these populations, which likely mean that these are potentially subspecies, and there's 3 subspecies of Hoiho. So that's what we're kind of working on now. We were obviously trying to get back to the main point of doing this was try and look for genetic disease risk. But also looking at the divergence times of these, our very preliminary analysis using Bayesian time-calibrated phylogeny estimates the divergence events between northern and southern populations to be about 3,000 years ago. And so this really could represent quite distinct subspecies. So this work is ongoing, funded by Genomics Aotearoa. So going back to our list of challenges that really hamper and make disease investigation in wildlife really, really annoying. Another example is something that was actually just published yesterday, and Stuff covered it with so many puns in the headline and the article that I actually just couldn't read it. But we were looking into this disease, this really important disease in Kakapo, otherwise known as crusty bum. And if you have never seen a Kakapo crusty bum, trigger warning, this is what it looks like. So there's this disease affecting the cloaca of Kakapo that has been around for a long time, but has since spread to multiple islands, which is becoming really concerning. Sorry, I'm going to get rid of that Kakapo picture. And the reason why I've been looking into it is because Doc approached us because it's really, they have to stay in the wildlife hospital for months for treatment with this disease. So it's become a really big problem, and the sort of causes have remained elusive for quite a long time. So we didn't find any viruses in this, which was annoying, but we looked at all the other species that were there. We found three really quite important looking bacterial species. And while we found a lot of species across, both diseased, healthy, and previous and ones that had previous infection, when we looked at the abundance of these, it became quite obvious that the diseased ones had really high abundance of these three bacterial species. And these bacteria are usually found in the gut, so the presence of them is nothing really shocking. It's just that they do have potential to be pathogenic, and at this level of abundance, then potentially it is contributing to disease. And when we looked into all of these to try and understand the sort of genetics of them, the E. coli is quite an obvious one here, where actually there's really low abundance levels of E. coli across all samples. But so we wanted to look at that a little bit more. And when we look at it at sort of a strain level or a sequence type level, we find this quite distinct pattern where all the diseased ones often had this subtype called 258. And although it's been found in birds and other things like possums in New Zealand before, it has really, we looked at expression of virulence genes and things, and it's got really high expression of genes that may make it pathogenic. So it's the first sort of time that we potentially have a leading suspect here. Again, the annoying thing about working with a really endangered wildlife like this is that basically you can't really do Cox postulates here. We can't do challenge studies and things like that. But turning it around and thinking about potentially having a very specific treatment, such as phage therapy or an antimicrobial that targets just this sequence type, could help prove causation in this case. So moving away from working with annoying wildlife to working with industry and primary industries, where we do often have model organisms or cell lines or potentially challenge studies that are possible. Been working with a few different industries to try and understand the causes of infectious disease in a range of different animals. The first is that we've been working with the Cawthorne Institute and the seafood industry to try and understand causes of tail fan necrosis. This is a disease that affects the tail fan, and it hugely affects profits of crayfish in the fishing industry, and it's been a really big problem. It's present in Australia as well, but there's been no causative agent identified yet. And so we again took this sort of meta-transcriptomics approach where we kind of sequence everything and just see what's there. And of course, it gives you a list of suspects. We looked at bacterial and other species. the only species that kind of stood out in these diseased ones, so these were sampled around Nelson Way, was this virus, and it's called our red rock lobster crustavirus that we named it. It's a novel virus. Sits in this clade with other viruses infecting crabs and other things like that, crab-like things. Its genome is quite strange, where it doesn't have a, oh, sorry, I do, it, sorry, it doesn't have a glycoprotein. And glycoproteins in viruses generally help with cell entry. So whether or not we can identify glycoprotein through sequence homology, or potentially it doesn't have one, there are viruses in this clade that don't have glycoproteins. So we don't know how it gets into the cells. So we don't know anything about this virus. We know it exists in these diseased lobsters. But what's kind of cool about this is that the Cochrane do husbandry trials with things like lobsters, and we were able to potentially do challenge studies and understand potential association with disease. So that's something that we're currently working on. Another industry partner that we've been working with are in farms, and a big one is cattle. So I read a statistic the other day that New Zealand has 10 times the global average of livestock density compared to the rest of the world. And I think looking at pathogens in livestock, it's really kind of opened my eyes into how much there is that we don't know, and how, these mammalian species that we work with quite closely and consume, how much they're sort of harboring, that seems to remain kind of a black box for us. So we've been looking at this disease outbreak that MPI investigated but couldn't really get to the bottom of with their sort of PCR-based tests. It included mainly oral lesions in the mouths of cows, and they weren't really sure what was causing it. But these cows were from a farm in Taranaki, and they had a range of symptoms. So often they would have oral lesions, often they would have oral lesions with diarrhea, and sometimes they even had diarrhea alone. And we got as many healthy cows as we could, but often they would then go on to develop symptoms. So we only ended up with 2 healthy cows out of the whole farm that we sampled during this time. So this was a collaboration with MPI, and who knew there were so many viruses in cows, but there was a lot of viruses, and a lot of viruses that were infecting these cows that we knew that could cause disease in cows, but don't necessarily have to cause disease in cows. They were viruses in healthy cows, and we were trying to grapple with the fact that there was differences in symptoms and lots of viruses at different abundance levels as well. And so there was this really complex network of transmission between the cows too, that involved the healthy cows as well as lots and lots of viruses, and their abundance. And so trying to understand what were the causes of these disease, if any, was something we were trying to untangle. We use this sort of differential expression analysis where we looked at the abundance, because abundance usually means viral load, which might mean pathogenic viruses, or at least a contributor to disease. And the only 2 that really stuck out were in the oral lesions. We had this parapox virus, which is related to pox viruses, and which is known to be in New Zealand already. And in the fecal samples, we had this novel virus, actually, Bopi virus, which wasn't known to be in New Zealand, but is known to infect livestock all over the world. So we think that these 2 are probably suspects that probably warrant further investigation. But again, this is something that's just another example of how genomics can be very frustrating, but yet somewhat useful. So as we prepare for this next threat of H5N1 and this subclade that is currently sweeping the world, that is likely to cause catastrophic damage to our wildlife, and they're already dealing with lots of infectious diseases, what are we doing, to prepare for this by using genomics? Well, we know that there are lots of entries for this pathogen into New Zealand, likely through migratory birds. So New Zealand, although we like to think is geographically isolated, we're really highly connected through these migratory birds that go up to the northern hemisphere and down to Antarctica, and thousands make landfall in New Zealand every spring. So the virus is also, as I said, on every continent apart from Oceania. So it's on the mainland of Antarctica. And of course, we have these stepping stones of subantarctic islands all the way to New Zealand. So whether or not it comes from the north or the south is yet to be seen. So I think it's a matter of when this virus arrives, not if. But what we can do and what we are trying to do is trying to use genomics to try and understand the risk and potential pathways of dissemination if the virus does get here. So we have a project where we're sampling lots and lots of wild birds, wild aquatic birds. This is primarily with DOC and lots of other researchers that we're trying to piggyback with. And this sometimes involves sending my postdocs down cliff faces in Sinclair to sample birds. And we're taking lots of swab samples from their cloaca and oral samples to try and understand these dynamics of what viruses they have, particularly influenza viruses, but other viruses too. So we've sampled lots and lots of birds over the past 2 seasons. We've just finished our second season. The birds have now left to go to usually the Northern Hemisphere for their summer. And these are all the sites that we've sampled, including all the main migratory sites that are really important for flyaway sites in New Zealand, including all the subantarctic islands and Chatham Islands as well. And we've taken quite a few thousand samples so far in this. We haven't detected high path flu. We've detected low path flu though. For example, in red knots, which are known to carry low path flu, it's nothing too concerning, and they were sampled right at the top in the Firth of Thames. And looking at the genomics of this virus, if we look at those really important surface proteins that determine the subtype, for example, in this case, it's H1N9, you can see that these viruses sit in clades with other viruses sampled from both Australia and Asia, which is to be expected given their migratory path. But you can see this sort of really long branch, particularly in this H9, which demonstrates that it's probably not a new introduction to New Zealand, but how little surveillance is done in New Zealand on wild aquatic birds more generally, and how little we know about the dynamics of this virus and these viruses generally in New Zealand. So it's really important to continue surveillance like this to try and understand those dynamics. And as I said, New Zealand is basically the terminus, the endpoint of this really important flyway site for migratory birds that go right up to the Siberia and land in New Zealand every year. And they pass through and land in places that have got really high prevalence of high path flu. So as I said, I think it's a matter of when this virus arrives here and not if. But what we're doing, I guess, is to try and look at all avian viruses in these migratory and sedentary birds as well, to try and understand virus sharing among species and potential dissemination pathways if a virus was to land at an entry point and how it would spread into New Zealand. So using this genomics-informed approach to try and potentially predict pathways of spread as well as diagnose them. So that's kind of ongoing work at the moment. So again, maybe ask me in a year how's that's going. So just to end, thank you very much for listening. There's lots of funders associated with all these projects, and particularly Genomics Aotearoa. All this work is mainly done by my lab. This is my lab group, and we're all women, which is great. And they've really led all this project. I basically do emails. So it's really up to them. And thanks to all the collaborators. Thanks for listening.
Here we go. All right. Thank you very much for a fantastic talk.